Oh, here we go. So that only took 15 minutes to get the projector up. We are talking today about embryological development. This is the last little content piece that we're going to have this year. So we just finished a whirlwind tour through ecology so that I could talk to you about dominion, and that was really the point of that. And now I do want to show you some cool embryological things with chicken embryos, and so I thought I'd at least teach you quickly what the science behind that is. So this is lifted out of chapter 25, but it's not all of chapter 25, it's just a little couple sections out of it. So embryological development, there's a lot here. Animals differ in the kinds of reproduction that they employ, but all embryos, whatever the environment, go through the same stages. So fish embryos go through these stages, worm embryos go through these stages. Every kind of animal goes through these stages of development. And so there's the stages we're going to talk about apply to everything. The pictures are going to be human because chapter 25 is about human embryological development. But these terms can be applied to anything, including the chickens that you will be dissecting on Tuesday next week. Okay? So there are three main strategies of sexual reproduction, at least within animals. These obviously don't apply to plants. But sexual reproduction in animals, you can be either oviparous, which means you lay fertilized eggs which hatch later. And most animals that are oviparous don't have much parental involvement. Most of the time, the parent lays the eggs and walks away. There are obviously some exceptions to that. Chickens are oviparous. They lay an egg that hatches later, but they also care for their young. And birds, generally speaking, care for their young. But fish and insects and most reptiles lay an egg and forget about it, right? And then the babies hatch later and fend for themselves. So most of the time, oviparous doesn't have much parental involvement. Birds are an exception. Then there's ovoviviparous, which is strange. It's where you have an egg, and the egg develops inside the mother and hatches inside the mother. And then babies come out, and so do shells. And so this looks like live birth because the mom gives birth sort of to babies, but the babies weren't attached to the mom's body in any way. Like there was no placenta, there was no blood-on-blood contact. The egg was just hanging out inside mom, right? And so lots of reptiles, some fish go through this strategy, and there's a lot of snakes wind up being ovoviviparous, where the egg is stored inside the mother and then hatches inside the mother, and then babies come out, and so do eggshells. Okay, so that's the in-between strategy, and then there's the strategy of mammals. And mammals are the main flagship group for viviparous birth. So you've all taken Latin. You're in your last week of Latin, so you can help me understand all of these words. Ovo, what does that refer to? Ovo, or ovi. Which means what? Which stands for what? Interesting. No. Eggs.
Oh yeah.
Yeah, an egg. Yeah, ovo is an egg. And
we didn't learn that
vive or vivus, or what does that mean? Vivi is life, right? Yeah. So this is egg bearing. This is egg live bearing, right? Because they're an egg, but then they come out like live birth, right? And then this is live bearing. So this is where the animal, there's no egg involved. So mammals do that. There are some other animals besides mammals that have viviparous birth, but mostly it's mammals. Most of the time, if another animal looks like it's giving live birth, there was an egg in it. Okay, so those are the three main strategies. Mammals obviously are viviparous. So this is oviparous, little chick. Hi, chickie. Okay, we'll see you Tuesday in pieces. And then this is the a snake, and this particular snake is obviously oviparous because there's eggs, but lots of snakes are ovoviviparous. So and then obviously turtles are oviparous as well. This is a snake that's ovoviviparous, where the snake is born as it's hatching out of the egg. There was an egg inside the mom. And then this is an example as well of ovoviviparous. These little baby stingrays.
Whoa,
Oh,
they're staring at us.
These little baby stingrays hatch as they emerge out of mom.
Is that bottom one? Pretty awesome.
And then live birth, obviously.
It just drops from that far.
Oh.
You're calling?
I'll spare you the pictures of human live birth. Okay. Okay. So
Wait, I never got three.
stages of development. This is the main thing that I wanted you to get. So obviously fertilization is the first stage. Egg and sperm fuse, and you all by this stage in your life know how that works, right? If you don't, ask Dad. But egg and sperm fuse, and then the partial nuclei from each gamete combine to make a new genetic individual, a new life. And that one fertilized cell is called a zygote. So the zygote is the word that we use for the one cell stage. And every sexual organism has a zygote. We even call the fertilized plant a zygote. Fertilized mosses are zygotes. So the first stage, only one cell is a zygote, a fertilized egg and sperm. And obviously we've already covered genetics. It's been a while, but in the human there are 23 pairs of chromosomes, right? And so you got 23 chromosomes from mom and 23 chromosomes from dad. And the two half nuclei fuse to make a new you with 26 chromosomes. Okay? Fertilized egg is called a zygote. Many of you are done writing, a couple of you are not. Pause. So here's a picture. This is the moment that you started to exist. This is an egg, this is a sperm, and that's the moment when this guy won the race. And this picture was obviously set up and taken so that there would only be one sperm, because the majority of the time it's not just one sperm that finds its way to the egg, it's whole bunches. Right? So, and the one that is the first to pierce this jelly layer on the outside of the egg cell will be the winner. But there's lots of competitors, right? So the one sperm that made it, he deserves the trophy. Oh wait, you're the trophy. He got to make you. So one sperm and one egg forms the zygote, the one-celled stage of development. This is what a human zygote looks like. And this is actually about to not be a zygote anymore, because you can see that the nuclei is already beginning to divide. So this is the one cell stage, but it's starting to become the next stage. So I'll let you write that down while I take the call. So for several days, the zygote goes through a rapid series of mitotic divisions, meaning it keeps dividing itself. And we call that stage, or that process, we call it cleavage, which just means to cut in two, right? And so it's one cell, and then it gets cut into 2 cells, and then cut into 4 cells, and then 8 cells, and up the powers of 2 it goes. Until it gets to the 16 cell stage. And at that point, we call it a morula. So morula is the name for a 16 cell ball of cells. The morula is fascinating because at that stage, every cell is specialized to the type of tissue that it will turn into. So the zygote can produce all kinds of tissue. By the time you have a morula, each of those 16 cells knows what part of the body it's going to produce. And so you have one of those cells that is called the ball on making skin and connective tissue. And you've got another cell that has said, I'm going to be responsible for all nerves. You've got another cell that says, I am the root of everything muscular, right? And so each cell has become, has started to become a type of tissue. It's incredible. At that stage, it hasn't gotten any bigger, though. So the zygote doesn't grow. The zygote stays the same size. It just gets cut up into 16 pieces, which is why we call it cleavage, because it's the same size, it just gets cut up into pieces, okay? So the zygote is as big as it is, and the morula, it has the same amount of mass. But the morula is 16 tiny cells, and the zygote is one really big one, okay? So here's what it looks like. The zygote actually leaves the same like jelly coat around it that it had as an egg for a while. So that jelly coat stays there. It goes to 2 cells, 4 cells, 8 cells, and then when it gets to 16 cells, we call it a morula. Okay, so all of those cells packed in a jelly coat. So this jelly coat that was on the outside of the egg remains. And here we have a whole bunch of cells inside. Each of those cells knows what it's going to become. It's pretty incredible. Okay? The morula now has to grow. And it puffs up. It goes, it's like roasting a marshmallow, only there's no marshmallows or fire. So it's not like roasting a marshmallow, but it looks like it. Where the morula goes from a solid mass of itsy bitsy cells to it like pops, it puffs, it gets bigger, and when it does that, we call it a blastula, and I always remembered that because it's like a popcorn kernel, blast. It gets bigger all of a sudden, poof. And when it does that, it goes from a solid mass to a hollow ball. So picture, if you picture like a ping pong ball. The ping pong ball is hollow, and it's just a little bit of plastic on the outside of the ping pong ball. If you were to melt that ping pong ball down to just a pellet of plastic that it took to make a ping pong ball, that pellet would be a lot smaller than the ping pong ball, right? And then if you were to picture that little pellet of plastic turning into a ping pong ball in just a matter of hours, it would look like it got so much bigger and it grew, but actually it just went from a solid pellet to a hollow ball. Can you picture that in your mind? And that's what happens here when it goes from morula to blastula, is the cells go from a solid mass of little cells to a big ball of somewhat larger cells. So now it's hollow, called a blastula. The cells migrate from their place on the morula to their place on the ball based on hormones secreted by regions of the embryo. This is incredible. As the solid pellet of cells, the morula, is turning into a hollow ball of cells, the blastula, there's a cell that says, everything associated with the head, come over here, and it secretes a head hormone. And everything that's going to be needed for the head migrates to that part of the blastula. And then there's another one that says tail region, I'm over here, and it secretes tail region hormones. And so there's 2 cells that are organizing everything else and secreting hormones for head region, tail region. And the cells from the morula find their place on the ball based on which hormones they're supposed to hear and how loudly they're supposed to hear them. If you're supposed to be in the middle of the animal cell, well, you want to hear the tail hormones about as loudly as you hear the head hormones. If you want to be a tail thing, then you go to the tail. If you're a head thing, you go to the head, right? So these cells arrange themselves based on the organization created by hormones that are secreted from regions of the embryo. And then these hormones are controlled by a group of genes called Hox genes. And you can take entire classes in college on Hox genes because it's incredible what those things do. It's like a master switch if you've ever looked at a power box, like a breaker box. It's a master switch that turns on or off a whole bunch of other genes. And those Hox genes say, I am going to make a leg. And cells in that region are like, okay, we're all about a leg. And it secretes hormones that say leg stuff. And so the cells in that area are like, okay, I've got all of my DNA here. I can turn off everything associated with livers and hearts and kidneys because I'm not going to make that. And then leg, need those genes. I can turn off all of the genes about whatever else. And I just need to look at this set. And so those Hox genes and the hormones associated with them are what tell the cell what it's going to become. It's pretty awesome. It's pretty awesome. And you can do all kinds of funny things where you take an embryo, not from a human, take an embryo from something else, and bathe the wrong parts of that embryo in the wrong hormones. So take a region of the blastula and bathe it in hormones to make a leg where it should have made it like a rib or back or stomach or something, and then that embryo will grow a leg there. Because it's like, you said I'm a leg. I'm going to turn on the leg genes and I'm going to make a leg. And you can see pictures of where people have done this with frogs with like legs coming out of the middle of their forehead and things like this. Like, it's crazy. So Marula, solid ball of cells, Blastula, hollow ball of cells, and the process here goes from, this picture doesn't really help you understand, but Marula is small, Blastula is big. So it really gets quite a lot larger when it goes through that process. These are pictures of Blastula. So hollow balls of cells, this is what a blastula would look like under a microscope. And the cavity in the middle we call the blastocele. Latin for a cavity or a space is cele, C-O-E-L. So blastocele is the space, the cavity in the middle of the blastula. Okay, we're almost done. Now, the blastula has to go from one layer to 3 layers. And the 3-layer stage is called a gastrula. So blastula and gastrula sound sort of similar to each other. I remembered it because blastula like blasts off and pops up into a big ball, right? And then gastrula, this is the stage where we have the gut for the first time. At the gastrula stage, I can point to the intestine. And so it's like the gastrointestinal tract, right? So the gastrointestinal tract shows up here. And so the gastrula, this is where you can have a gut. And so the blastula puffs up into a hollow ball, and the gastrula is the 3-layer stage. Now, when we do this, what happens is the solid ball gets kind of like a belly button. It gets a point where it starts to pooch inward. If I had a balloon, I should have blown up a balloon for this demo, I'm sorry. If I have a balloon, picture a balloon, and then you poke on the balloon and you push the rubber from one side of the balloon through the cavity and make it touch the rubber on the other side of the balloon. Like you just push your finger through and you're making the balloon poochy inward on itself. The poochy inward part, depending on which species, the part where it starts to poke inward on itself might become the mouth or it might become the anus. And there's different groups of animals based on does that hole become the mouth or the anus. And so there's a word that means mouth first. Protostome means the first hole. Proto means early, stome means hole. So this is protostome, the first hole becomes the mouth. Protostomes are animals where the first hole, where the blastula starts to pooch in, becomes the mouth. The other strategy is where the first hole is the anus, and as the poochy part makes its way across to the other side of the blastula, it pokes through the other side and makes a second hole. And now you have a tube within a tube. And if the mouth is the second hole, then that's the deuterostomes. Deutero means second, stoma means hole. So is the mouth the first hole? Well, then you're a protostome. Is the mouth the second hole? Well, then you're a deuterostome. Okay? So that results in 3 layers because you have the outer part that has always been the outside. You have the part that was just created as you poochie through it, right? Okay? And then the part that poochie through it, now the tube that's inside the sphere, starts to pooch again and form another layer of stuff inside. So you wind up with 3 layers. And the outermost layer is called the ectoderm. Ecto means outside, derm is skin. And that becomes the skin and the brain. And then the mesoderm, the middle part, the middle skin, becomes the internal organs. And then the endoderm, the inside skin, becomes the gastrointestinal tract. Let me show you some pictures here, but first